Welcome to NPTEL online certification course on Pharmacognosy and Metabolic Engineering. This is lecture 55. So, where I will discuss metabolic engineering of anthocyanin pathways in flowers. So, in lecture 54 I have discussed in details about the anthocyanin biosynthesis, the chemistry, structural diversity, how simple chemical transformation, hydroxylation and methylation can convert one anthocyanin into another, their stability and how they are transported into the vacuole, and as a very exciting metabolic engineering application with expression of transcription factors in leaf, leading to produce huge amount of anthocyanins in tobacco cell cultures. So, now here we will talk about the metabolic engineering attempts made with the flower. So, this has tremendous industrial application because ornamental plants there is a very huge market, and as you see that there are multiple colored flowers are coming, but even with the traditional breeding everything cannot be achievable, particularly the blue coloration is a challenge, like if you can make blue rose, blue carnation. So, there is a huge market. So, unfortunately, the achievements was not that much, but still there are a lot of hopes there. So, scientist has cracked the major bottleneck, and I think years to come we will be able to see blue flowers, blue rose flowers. So, now go to the next slide. So, the concepts to be covered: biosynthesis of anthocyanins I have already discussed, manipulation of anthocyanin pathway, that is our concept, that is our theme of discussion in this class. So, I will talk mostly about that yellow flower by redirecting flavonoid biosynthesis. So, here and then making blue flowers and then attempts to make rose with blue hues. So, again this scheme, now I have mentioned this scheme several times. So, I can avoid. So, anthocyanins are C6, C3, C6 structures. So, and this scheme also I have discussed in the previous class in pretty details. So, only thing to mention here that these are the target genes which need to be either up regulated or down regulated, leading to the formation of enhanced or suppressed anthocyanin accumulation. So, CHR is a new one. So, CHR will come in due course. I will talk about CHR. So, if you remember CHR I have mentioned particularly in the context of isoflavonoid pathway, where CHS and CHR work together and which produces ultimately the isoflavonoids. Okay. Now, modification of anthocyanin amount. So, one target was set to make basically the brick red or orange red pelargonium. So, transgenic petunia. So, petunia colors, brick red or orange red petunia colors are rare. So, scientists what they have attempted, basically they have attempted to hire maize dihydro DFR, dihydroflavonol reductase. So, a maize DFR to be transferred into the petunia and to see whether it makes any effect on coloration. So, that is one, or also the F3 prime H, F3 prime 5 prime H can also be targeted. So, what we see as a expression of CHS and chalcone synthase in petunia, mini purple plants, resulted in few plants with pale color or pure white color. So, this is antisense. So, when means that when you block the chalcone synthase, that is the first step. So, that means you are blocking the entire pathway. So, coloration will be theoretically suppressed, and in practice also it happens. And but sometimes some transcription factor genes like maize R genes, when they are transferred in tobacco, that making more intensely colored pigments, because that transcription factor has some role in upregulating the anthocyanin pathway. Similarly, antisense suppression of flavonol synthase gene in petunia also resulted in higher anthocyanin content. So, that aspect maybe will come later. So, here is basically the modification of flower color in petunia by expressing DFR, and DFR is from the maize, and as a result of that what we see that the color of this turn different. Okay. So, transgenic cultivars express the maize DFR gene which is showing here, and B1 is the red flowering petunia cultivar that accumulates cyanidin type of anthocyanin. So, so this is basically the DFR, and the DFR basically what it converts dihydrocampherol to this eventually to the pelargonidin glucoside, and if it accumulates it makes this color. So, okay, and so this is reported long back in Trends in Biotechnology, and this is another one where the bottom 4 are as a result of transgenic application, whereas A to D, that A, B, C, D, these are basically non-transgenic, maybe it's created out of the mutation, but whereas this is all, these are also petunia. So, modification of petunia color by genetic engineering. So, this was also reported long back in the famous journal Plant Cell. So, here basically in case of E, if you see, now we can see one by one. So, this one. So, here what happens, the antisense CHS, when I mentioned this just now, that antisense CHS blocks the entire pathway almost. So, no coloration leads to white. Now, second one is basically they transformed a rose DFR gene, and as rose DFR gene basically rose is basically cyanidin. So, the DFR converts what the dihydroquercetin to leucoanthocyanidin and subsequently to cyanidin, and as a result of that it is developing the red coloration. Okay. And the third one is basically they have transferred a gene which suppress both F3 prime 5 prime H and flavonol synthase activity. So, as a result of that what happened, that dihydrocampherol will mostly accumulate, and that dihydrocampherol may leads to the formation of pelargonidin, and as a result of that you will get this orange red color. Okay. And the fourth one is basically transformed with a sense petunia F3 prime 5 prime H. So, when petunia F3 prime 5 prime H is transferred, then what will happen, that that leads the pathway flow more towards the delphinidin, but if the background is with if the plant transgenic plant, the explant used is already containing another gene of say which led to the formation of reddish color, then with the blue color what will happen, these two will mix up, and as a result of that there will be different coloration. So, if you see these three, these have 3 different coloration, those strategies are different, one antisense, one overexpression, one another gene. So, as a result of that, if one play with these pathway genes, it is possible to make the changes in the flower coloration. So, this one I skip it. Now, come to very important and interesting thing, like normally what is happening, malonyl CoA joins with 3 molecules of malonyl CoA joins with 1 molecule of paracomodyl CoA, and that converted into tetrahydroxychalcone, and the enzyme is chalcone synthase CHS. However, as I said, in case of this isoflavonoid pathway, so there CHS work with CHI, but there CHI is not there, but instead CHR is there. So, in other word, in this case a pathway will move in this direction, where CHR and CHS will work and leading to the formation of 6 prime desoxychalcone. And this is not very clear here, but this is 6 prime desoxychalcone. Now, what happens that 6 prime desoxychalcone is not the natural chalcone that accumulates in the flower, okay. But it is possible how, if you take a Medicago sativa, which is basically a legume. I mentioned try to find the relationship. What I said, the legume produces isoflavonoids and isoflavones, and there the CHS work with CHR. So, that is why scientists they have hired the CHR from Medicago sativa, okay. So, and that they express in petunia. So, what happens in this case, the so the pathway now will moves in a new direction, and eventually the CHS work, CHS will make this tetrahydroxychalcone. That tetrahydroxychalcone cannot really converted into naringenin. Instead, what I said, the pathway will move in this direction, okay. And then what will happen, that this leads to accumulation of 6 prime desoxychalcone, and this is possible if you express the genes under the control of a strong promoter. There will be more CHS as compared to CHI. So, therefore, the whatever this tetrahydroxychalcone will be produced, that will be channelized towards 6 desoxychalcone because of the new enzyme which you have introduced. And the beauty is this is a new compound and it is unnatural. So, the petunia cells cannot metabolize it further. So, it started accumulating. Once it started accumulating, what it will see, what you will see? You will see the accumulation of yellow coloration. So, accumulation of yellow coloration. Okay. So, this is what is called redirection of flavonoid biosynthesis. So, flavonoid biosynthesis normally moves in this direction. Now, you are blocking that and allowing this to move in this direction. So, that is how it is called redirection. Now, let us see the result as a result of that. So, they have used two type of petunia, one is colored, one is white. So, white is acyanic, not cyanic means colored. So, acyanic is the one you see, this is C, left is the control, this is control, okay. And then they have expressed the genes, and as a result of you see the changes in the coloration. So, this is control. So, if you, and this is the test transgenic, this is the transgenic. So, what you see that you compare to this and this, the yellow coloration is noticed here. Here yellow coloration is quite intense, and here actually 3 developmental stage, the left one is control. So, this is a control and these are the transgenic. So, the products, so the yellow coloration it is possible to produce in a acyanic background, that is the white petunia if you take, and then if the gene is transferred and expressed properly, then this white petunia will produce upon gene expression will produce the yellow coloration. But if you take the cyanic background, then what will happen, that already it has a color. Now, when you have done the paint brush work, means with paint and brush in a paper. So, when you a color is red is there. So, you add a yellow, what will happen? The original color will become diluted. So, similarly, that is what is noticed here, and so the color, the original intense color will get diluted. As a result of that, you will get different pattern of coloration here, and only thing is that I will mention here the when it is redirection, that means more of chalcone is produced, isn't it. So, what you see here, the chalcone percentage in this line is 51.7, chalcone percentage is this line is 62.5. Okay. So, that means more chalcone is basically producing, more chalcone is accumulating. When more chalcone is accumulating, so it is producing yellow desoxychalcone. So, that is basically mixing up with the other anthocyanin, and as a result of that, if the pH is favorable, as a result of that it makes different pattern of coloration, and some of these colorations are indeed novel, which cannot be achieved by traditional breeding. So, other example is this metabolic engineering to modify flower color. One is the petunia, they have used another variety, purple variety, and they use antisense suppression of chalcone synthase, and as a result what you see this is the control which produces this color. When you suppress, depends on the level of expression, you see that coloration changes and eventually it produces white flower in different lines, depends on the expression levels, how many copies of genes inserted, all these will influence how strength is your promoter. And the second one is this, that here basically they have transfer petunia or rose DFR, and as a result of that what will happen, the novel flower color in the left side, this is the transgenic and this is the no, this is transgenic, sorry, this is the control. So, the novel coloration was developed, as a result of that the rose, when the rose DFR was expressed in petunia. Okay. So, this is a novel color. And third one is this, that here the this is the control. It normally produces this bluish color, this is Torrenia which produces bluish color, and this is a summer wave, a particular variety. And what they have done basically, they basically made the sense suppression of the CHS or DFR. As a result of that, you see that which is a more bluish purplish color, now it changes its coloration. So, some portion is white, some portion is bluish. So, that means a novel coloration pattern developed as a result of sense suppression of CHS and DFR genes. So, and this was reported in some of these were unpublished those days when this review was published, from there I have taken. So, this is fine, but ultimately what I said at the beginning of the course is that beginning of this lecture is that eventually we aim to make blue floweration, blue color. So, if you can make novel blue color, particularly in this carnation or in this Torrenia or in rose, you will have a very good market, horticultural market. So, but in order to produce blue flower, it has to meet several conditions. One is the synthesis of 3' 5' hydroxylated anthocyanin, that is required, that means delphinidine. So, 3' 5' H activity must be there, and also there is a need for presence of flavonol copigments. So, flavonol copigments are required. For example, that delphinidine also you also require the myricetin to be present there. So, when myricetin to be present, so that is basically the flavonol copigments. So, that will work and in with the delphinidine and makes the stability of the coloration. And another important point is that I have mentioned the high vacuolar pH. If you remember previous one of the previous slides, I have shown that blue coloration means between 4 and 5. So, that is important. Now, flower petals are actually meant to have lower pH in order to make the stable anthocyanin. So, making a higher pH is also a challenge, and then also if 3' 5' H activity, acyl transferase activity is also necessary for producing the blue color. Now, what has been achieved is this, that this is the requirements, and this is the achievements and the bottlenecks. Achievements is the genes encoding F3' 5' H has been isolated from petunia, eggplant and many others, and a transgenic violet carnation was successfully developed by Florigen Limited and Suntory Limited, both are this one is Australian, another one is Japan based companies. So, where they have introduced a Petunia F3'5' and DFR gene into a DFR deficient white carnation. So, white carnation is where is the DFR deficient, that means DFR genes are not working there. So, that is why the if DFR is not working, then you know what is happen, dihydrochemferol will accumulate. So, if only dihydrochemferol accumulates, if it cannot move towards pelargonidine or towards delphinidine or towards cyanidine, what will happen, the color of the flower will be white, because it stops there. So, they have taken a DFR deficient white carnation and there they have introduced F3'5' and then as a result of that they perhaps they have also had another DFR from other source, and as a result of that they see changes in the coloration. Now, now apart from these are the some achievements, but still some bottlenecks are still there, that is the control of the vacuolar pH. So, vacuolar pH only few genes have been cloned. So, which really maintains the proper vacuolar pH, so that you get the stability of the coloration. And so, this is what is that the mouth coloration developed by introducing a HF1 and DFR genes in white flower carnation variety moon dust, and which I said that why and that accumulated dihydrochemferol. So, when DFR was used, so here dihydrochemferol accumulates, and what happens that this one normally dihydrochemferol if accumulates it makes white color, but here particularly when they have hired a DFR from different sources, this basically leads to the formation of a novel color in this carnation. Okay. And next is this is the transgenic violet carnation produced delphinidine type of pigments, and this is because they have transferred F3'5' and there are two, the left one is the paler one. So, this one is the paler one which is called moon dust, and this one is the darker one which is the moon shadow, and this is the moon dust. And with further advancement it was possible to make some blue hues in this carnation as well as in rose, which was not possible by traditional breeding. So, next attempts were made by this Suntory and Florigen group, they worked together and they really aim to make blue colored rose. So, blue colored rose, as you see from the knowledge what we have gained, that in order to have blue colored rose, the delphinidine must be accumulated. So, if delphinidine to be accumulated, then F3'5' should be worked, and along with that the myricetin, myricetin is flavonol pigment. So, myricetin accumulation is also required, okay. And apart from other. So, native rose only accumulate pelargonidine and cyanidine based anthocyanins, that is so what is this? So, either pelargonidine or cyanidine based anthocyanins. Okay, and delphinidine based anthocyanins, basically there is a deficiency in normal rose of F3'5'H, that is if it is there at all, it is non functional. So, it is important that the F3'5' to be hired from a heterologous source, so that, and if it is expressed, so that dihydroquercetin is a common one. So, the normally dihydroquercetin for cyanidine it moves in this direction or this is, but then with this the pathway should be able to move in this direction, and as a result of that, if the rose petal generates delphinidine, and again there are lot of factors, only delphinidine will not help, and along with that myricetin should be produced in higher amount, along with that the stability pH should be checked then only. So, therefore, gene is available. So, therefore, in order to have a target rose cultivar where the gene to be transferred, it is important to do a thorough screening to find out a rose cultivar where a rose cultivar accumulates more flavonol as co-pigments like myricetin. So, with screening it is possible, with screening of the flowers of hundreds of cultivars, it is possible to pick up couple of 3, 4, 5 rose cultivars which are rich in myricetin flavonol. So, that is also another target, and next target is that to check the vacuolar pH, petal pH, to check where the pH is in higher side. So, with their thorough screening, the scientists from these industries, they are able to find out at least 10 rose cultivars which actually speaks in favor of their work. That means, where myricetin content is more, along with that the vacuolar pH was in the higher side. So, that was important, and now what they have done. So, now they have made different constructs. So, one construct is overexpression constructs, where F3 prime 5 prime H they have hired from viola. Viola, the name itself is that it produces violet color. So, when is a violet color, then F3 prime 5 prime H is there. So, that they have hired there, and they have hired another acyl transferase from Torrenia. So, this construct they call PSPB 130, and when this construct was expressed in rose, what they found? They found changes in coloration. So, the left is host, right is transformant. Host means we can write control, host, okay, host, and this is transformants. So, you see changes in the coloration. So, when F 3 prime 5 prime H is expressed, the deep red color, is now getting diluted towards more pinkish. Here also it is getting in this color. Okay, here is slightly different, difficult to explain. Here it is more towards bluish. Okay, and here also it is quite clear, but not everything cannot be explained. So, this is what they have published. They have published everything in this particular paper. So, we will come to that. So, this is one. So, they got some success with this. So, next what they have done, they also attempted to make only with a viola and see, but that result was not that interesting. Next what they have done, they have used iris DFR and viola F3 prime 5 prime H, 2 genes together. So, that gives a better one than the previous, but what happens next? What they thought that the main culprit was the rose DFR. So, what happens even if you put the DFR and, so what I said here, that what you are hiring a DFR, you are hiring a DFR and 3 prime 5 prime H, but what happens, okay, you are hiring a DFR, this one, sorry, I will erase this one, not DFR. What will happen, that this particular DFR is already there in the rose. So, when you are hiring F3 prime 5 prime H and DFR from 2 different plants putting there, but these DFR which basically converts dihydroquercetin, which is the major intermediate, from there only it converts dihydromycetin. If this DFR is strong, the red one which I am showing here, if this is have a more stronger expression, so what will happen? It will try to pull the substrate more towards cyanidin. Even if you put extra copies of F3 prime 5 prime H and DFR from different sources with expression, the endogenous DFR will not allow it to function. So, the strategy what they have learned from this that the third strategy what they have done is that they have used a RNAi approach to block the rose DFR. So, the rose DFR to be blocked using RNAi, that is why I have shown in this way. This is basically a hairpin construct, and I think hairpin construct I have mentioned when I talked about the this morphine alkaloids. So, in that or that time, because now because of shortage of time, again I will not discuss that. So, you can always revise from there. So, now, so the number 1, let us see, first one is that they had hired a iris DFR, that is good. They have hired Viola F3 prime is good, and the third strategy is basically they block the Rose DFR. Now, situation will be favorable. So, that means the if why I say favorable, then this will not work anymore. So, if this will not work anymore, cyanidin formation will be significantly reduced. Instead, dihydroquercetin will be moved towards dihydromyricetin, because you have hired a Viola, what you have, you have Viola F3 prime H, and you have also hired a iris DFR, and as a result of that, the pathway will move in this direction, and eventually it produces delphinidine, and this, as I said, this is already producing in higher amounts. So, this plays an important role in stabilizing the coloration. Okay. So, this is the best construct. With these best constructs, what they have achieved. So, they have achieved a coloration like this. And where the 98% was delphinidine. So, the flower color is evidently bluer than that of the host, the commercial varieties of this violet, and this is regarded as the bluest of current rose varieties. So, you see this color, and some of the transgenic rose derived from this also exhibited paler color containing decreased amount of anthocyanin. So, what happened? This was the target, and that now produces this. So, this was the cultivar which is producing this sort of coloration. One thing you see, this coloration is not the, this is slightly paler red, this is slightly paler red. They have purposefully chosen such one, so that the red will not interfere, and that they have blocked the DFR, rose DFR, they have added the iris DFR and add the Viola F3 prime 5 prime H. So, that leads to formation of this coloration, and they claimed in this paper that this was the bluest so far achieved, and this is really an outstanding work, and this was published in Plant and Cell Physiology. So, after that I have not come across with further work, maybe because it is a industrial work. So, there is a confidentiality to be maintained and patent issues and other thing. Once those were sorted out, and definitely they will publish. So, maybe we have to wait for few more years when we will may see a deep blue coloration rose available in the market. With this I end this class, and this is all about metabolic engineering of anthocyanin pathway, and you see lot of positive results with this, and the beautiful science behind this, that also I have explained. With this I end this class. Thank you.